first step in DNA replication is to unzip the double helix structure of the DNA molecule. Dry erase keeps the two strands from unwinding completely. The result of this is the replication fork. The separation of the two strands of DNA will act as templates for making the new strands of DNA. One of the strands is oriented in the 3' to 5' direction. This is the leading strand. The other strand is oriented in the 5' to 3' direction. This is the lagging strand. A short piece of RNA called a primer comes along and binds to the end of the leading strand. The primer acts as the starting point for DNA synthesis. DNA polymerase binds to the leading strand, adding new complementary nucleotide bases to the strand of DNA in the 5' to 3' direction. This sort of replication is called continuous. Numerous RNA primers are made by the primase enzyme and bind at various points along the lagging strand. Chunks of DNA called Okazaki fragments are then added to the lagging strand, also in the 5' to 3' direction. This type of replication is called discontinuous. Once all of the bases are matched up, A with T and C with G, an enzyme called exonuclease strips away from the primer. The gaps where the primers are then filled by yet more complementary nucleotides. The new strand is now going to be proofread by an enzyme called nuclease. Once all mistakes are corrected, an enzyme called DNA ligase seals up the sequence of DNA into two continuous double strands. The result of DNA replication is two DNA molecules consisting of one new and one old chain of nucleotides. This would explain the term semi-conservative, because the new strand of DNA now has a part of the parental strand of DNA. Following the replication, the new DNA automatically winds up into the double helix.